Hello everyone, I am Amay Ajaykumar Agrawal and I am PhD student in Professor Olga Kalinina's lab at Helmholtz Institute for Pharmaceutical Research, Salem. Today in this video, I will give a brief overview about my poster. I will show you how we are planning to use explainable AI to tackle antimicrobial resistance. Now, as we all know, antimicrobial resistance is an urgent threat to the human health worldwide as the microbes have developed resistance to even the most advanced drugs. Whole genome sequencing, followed by rule-based analysis, have accelerated the AMR diagnosis as compared to the traditional phenotypic testing, but it cannot detect resistance caused by unknown AMR markers. Prediction approaches based on machine learning provide superior performance over these rule-based approaches, and this is the basic pipeline of how machine learning is used for phenotypic detection. Now, although these machine learning models have achieved superhuman accuracy, but these machine learning models may or may not be always interpretable, which limits the use in clinical settings. Thus, in our study, we want to go beyond this and we want to build inherently interpretable models that explains how it is arriving at a certain prediction. And specifically for our study, we are planning to use the pattern mining approach of explainable AI to discover subgroups. And next we will see with a small example of how we are planning to use our approach in our study. So we are all aware of this unfortunate Titanic tragedy where more than 1500 people lost their lives. Now, during those times, it was an unwritten rule of sea that in such life-threatening situations, women and children are the first ones to be saved. But was this rule of sea really followed or not? And next we will see how we can use machine learning to check whether the rule of sea was really followed or not during this time. So first, you collect data about uh, all the people that were present on ship during that time. Next, we extract important information about all the person like their nationality, age, weight, sex, etc. And finally, we have our target variable that we want to predict, which is survival in this case. That is whether the particular person survived during this tragedy or not. Next, we use all this data to train our machine learning model, and we hope that the model outputs something very simple like this. But in reality, the model uses some complex mathematical equations to arrive at a certain prediction. And so this is like a black box to us. Now, we want to go beyond this in our study. And now if we just focus on sex, and in sex also if we just focus on females, we see that there is 100% survival rate. So this is basically just a pattern. And in our study, we can have individual SNP as a pattern, and all the bacteria that contains this individual SNP forms a subgroup. Now these patterns can be of length 1, or we can also have larger length patterns like patterns of length 2 or pattern of length 3, or we can also have even more larger length patterns as we can see here. Now, after looking at our approach that we are planning to follow in our study, next we will look at the pipeline that we used in our study. So first step is the data collection, and currently we are using the publicly available data from the Patrick database. Next, we do the pangenome analysis to split our genome into core and auxiliary genome. This core genome consists of genes that are present in all the strains, while the rest of the genes belong to the auxiliary genome. Now, this step of performing the pangenome analysis is important in our study because for pathogens which have an open pangenome, most of the resistant genes are part of this auxiliary genome. Next, we use the multiple sequence alignment of individual genes from all the strains and use them to build the pangenome reference graphs. And further, we use this pangenome reference graph to perform the variant calling as we can see here. Finally, we use these variants as a feature to extract important patterns related to resistance using our explainable artificial intelligence model. We applied our models on publicly available AMR data for Mycobacterium tuberculosis against commonly prescribed antibiotics such as fluoroquinolones and aminoglycosides, and next we will look at the results. So for ofloxacin, which is basically a fluoroquinolone, we found this single length pattern which contains the mutation in this gyrase gene, and the length of the probability of resistance of this pattern is also very high. And this mutation in the gyrase gene is already well known, reported in literature for fluoroquinolone resistance. Next, we found this second pattern, which is basically a 7-length pattern, which apart from containing the already reported mutation in gyrase gene, also contains mutation in some other genes. And the most interesting part about this pattern is that the probability of resistance for this pattern is even more as compared to this previous pattern. So definitely this mutations in these other genes might be playing some role in resistance, and it will be interesting to see uh, which role they play. Further, we also found such patterns where there is no known reported resistance mechanisms, but the probability of resistance for such patterns are also very high, as we can see here. Similar kind of results were obtained for other aminoglycoside antibiotics like amikacin, kanamycin, and streptomycin as well. Finally, from this, we can conclude that we discovered most known common resistant mechanisms, and additionally, we also observed some novel sets of markers that can be related to resistance in rare cases. In future, the function of these novel markers can be determined and can be further used in AMR diagnostics. And to know more about this project, please scan this QR code.